Hello, my name is Ian Ong, and I'm a third-year medical student at the Perelman School of Medicine at the University of Pennsylvania. Today I'm going to be speaking about a truly revolutionary new field which is using cutting-edge technology to bridge the gaps between disciplines, cancer neuroscience. In this presentation, I will first provide some background and then present a whirlwind review of a few key papers from leaders in cancer neuroscience. Cancer neuroscience is a budding field that sits at the intersection of cancer research, clinical oncology, and neuroscience. It emerged from observations that, alongside development and maintaining tissue homeostasis, the nervous system plays a parallel role in the regulation of cancers. The CNS and the PNS can influence tumor progression, metastasis, and response to therapies. And this poises methods derived from cancer neuroscience as the next potential pillar in the treatment of cancers across diverse tissues. Neurons can interact with cancer cells in a multitude of direct and indirect ways. This can include paracrine interactions, neurotransmitters that alter immune system function, and so on. Here is a graphic from Winkler et al., which summarizes the numerous ways in which cancer and the nervous system can interact. First, panel A shows that neurons can synapse directly onto cancer cells, leading to direct electrochemical interactions. In panel B, as I mentioned earlier, paracrine release from neurons regulates the tumor microenvironment, and in turn, cancers can actually secrete their own signaling molecules to remodel nearby neurons and hijack these pathways. Panel C shows circulating neurotransmitters and neuropeptides and how these can have relevant systemic effects on the body, such as altering immune function. Panel D represents the three-way interaction between neurons, cancer cells, and immune cells, and how this can produce complex behavior. In fact, neuroimmuno-oncology is a growing subspecialty aimed at detangling these interactions. Panel E shows that cancer cells may leverage signaling characteristics classically associated with neural cells, such as autocrine signaling. And finally, in panel F, we can see that cancer therapies themselves can have effects on peripheral nerves and overall cognitive function. Here's a roadmap which contains a sampling of the new developments being made in the field of cancer neuroscience. Much of the research in this field has been directed towards glioblastoma in particular, which is a highly aggressive cancer and the most common primary malignant brain tumor. So first, we're going to review two papers which studied how different communities of glioblastoma cells had different propensities for invasion, as well as taking a look at autonomous rhythmic channel activity in glioblastoma cells. However, cancer neuroscience is also applicable to cancers found in the peripheral nervous system and even beyond, such as in triple-negative breast cancer and prostate cancer. To try and illustrate this, we're going to look at a study of tumorigenesis in neurofibromatosis type 1. And finally, we're going to discuss a paper linking olfactory sensory experience to gliomagenesis in a mouse model. Glioblastomas are incurable brain tumors thought to arise from astrocytes. Using longitudinal 3D in vivo imaging of patient-derived human cancer cells in mouse models, Venkataramani et al. Introduced a gap junction permeable dye called SR101, which was able to highlight two distinct groups of cancer cells, ones that were anatomically connected and ones that were not connected to astrocytes or any other cancer cells. And using two-photon microscopy, they found that these unconnected cells are the main drivers of diffuse brain colonization, whereas the connected cells remain mostly stationary. Finally, using in vivo calcium imaging, they found that these invasive glioblastoma cells leveraged calcium signaling from neurons to increase their invasivity in a tumor microtube-dependent manner. Here is a figure reproduced from the paper which shows examples of the in vivo calcium imaging, and this is illustrating the complex calcium channel activity which takes place across microdomains in the glioma cell, such as tumor microtubes and the soma itself. In this same vein, Hausman et al. Published a paper a year later which found that tumor microtube-connected glioblastoma cell networks include a small plastic population of highly active glioblastoma cells that display rhythmic calcium ion oscillations. They identified KCA 3.1, a calcium-activated potassium channel, as driving this activity. So using this as a target, They treated mice with KCA 3.1 inhibitors and found that it inhibited glioblastoma cell proliferation and overall tumor growth after just 14 days of treatment. And over here on the right, I've included a figure which gives you a sense of the calcium transients in the glioma networks that were studied in the paper. This is a figure which shows the effect of KCA 3.1 inhibitors on brain tumor growth. The first two plots, A and B, show a reduction in periodically active cells and global calcium activity, respectively. Then plots were provided to show a significant decrease in cell density, a decrease in tumor size, and an increase in survival for both Seneca-POC-treated mice and KCA 3.1 knockdown mice, with the latter showing a dramatic increase in survival rate and a p-value less than 0.0001, and that's in figure J. Now we're going to switch gears a little bit. Because of neurofibroma's intimate association with nerves, researchers have also tried to apply the principles of cancer neuroscience to neurofibromatosis type 1. Neurofibromatosis type 1, or NF1 for short, is a cancer predisposition syndrome marked by benign peripheral nerve sheath tumors that derive from preneoplastic NF1 deficient Schwann cell precursors. In 2022, Anastasaki et al. Found that NF1 mutant dorsal root ganglion neurons, which extend sensory axons to neurofibromas, exhibit greater action potential firing rates relative to wild-type controls in a mouse model. They are able to target the implicated HCN channel using lamotrigine, and this blocked NF1 optic pathway glioma progression in vivo. Overall, they found that NF1 mutations modulate tumor predisposition by increasing neuronal excitability and paracrine production. Here's the author's proposed model for NF1-associated growth of low-grade tumors as reproduced from the paper. Panel A shows a schematic in which the deregulation of the HCN channel in NF1 mutants leads to increased baseline neuronal excitability, thereby stimulating Schwann cell proliferation and neurofibroma growth through a pathway which involves COL1A2. Panel B for optic pathway gliomas invokes a similar mechanism, but also includes a separate mechanism which involves visual experience light-induced activity. And this enhances production of soluble NLGN3, and this molecule drives optic pathway glioma initiation and cell growth. So we just took a look at the relationship between optic pathway glioma and light, and now we're going to look at a paper which suggests that gliomas may even be regulated by neurons relating to smell. In 2022, Chen et al. Studied NF1 knockout mice, and they found that insulin-like growth factor 1, IGF-1, a paracrine signaling molecule, played a role in the development of olfactory bulb high-grade glioma through a sensory-dependent mechanism. They then took their mice, they used plugs to occlude the left nostrils, and they found that this suppressed the development of gliomas through an IGF-1 and olfactory sensory experience dependent mechanism. Using the results of this study, future research in humans could focus on brain regions that respond to external stimuli and IGF-mediated tumorigenesis. So they also took a look at gene expression. Figure A here represents the scheme that they used to screen for narrowseclusion-controlled gene expression. Then, in figure B, the heat map shows 17 genes that exhibited consistent downregulation after narrowseclusion. The images on the right represent immunofluorescence staining and Western blots of IGF-1 EGFP mice with left narrowseclusion. In figure G, you can see the downregulation of EGFP, and this is serving as a proxy for IGF-1, and you can see that this is downregulated in the left olfactory bulb following narrowseclusion. All right, so this was just a small sampling of some of the great work that's come out in the past couple of years in this field. In conclusion, recent advances in cancer neuroscience highlight untapped opportunities for novel therapeutics and approaches to management. Future research could focus on translational work and harnessing evolving technologies. For example, one could consider combining functional imaging techniques with spatial transcriptomics, and this could be used to identify functionally relevant genetic products. Additionally, retrograde tracing with advanced viral vectors could help to reveal maps of neuronal inputs. In terms of tumors arising outside the central nervous system, directions for research include using imaging modalities such as 2-photon and light sheet imaging to map innervation, developing assays to characterize tumor nerve crosstalk and anti-tumor immune responses, and studying how nerves physically grow into tumors and how tumors can remodel neural circuits. Here's a list of all my references. And thank you so much for listening.